And so with that, it is a privilege for me to turn the mic over to Jane Frankenberger, who will be moderating the session this morning.
Thank you, and it's a pleasure to be here this morning and talk about two concepts: conservation drainage and climate smart agriculture. It was hard to find an image that really conveyed climate smart agriculture. I took this one from the USDA Partnership for Climate Smart Commodities. I think it does a good job. And we know these two are related, but what we're going to discuss today is what that relationship means. Yes, I'm Jane Frankenberger from Purdue University. I'm saying organizer and moderator, and I'm also going to give a brief introduction here, so don't think I'm just taking all the time from these speakers, to why we're doing this session. Now, the concept of climate smart agriculture has two components. There's the adaptation, where we think about increasing agricultural resilience. I and many others in this room have done a lot of work with that. It was kind of the focus of the Transforming Drainage project, where we've talked about increasing water storage in the landscape. The other concept is mitigation, where we're actually talking about reducing greenhouse gas emissions. And this is the focus of this session. Our whole session, we're going to talk about mitigation. Now, people who know me well may be thinking to themselves, Jane doesn't know about mitigation, and she doesn't even know how to measure greenhouse gases. And you would be right. But I wanted to say that because I suspect that the same is true for most of you out here, right? This is a hard concept. We know it's very difficult to measure. And so I suspect many others, we kind of listen to all the talks about greenhouse gas emissions, but we don't pay that much attention because it's really hard. What we hope is this session is going to make things clearer, at least for everybody, no matter what your current state of knowledge is. So the first thing I would say is we have a great panel, and I'm going to come back in a minute and introduce each one of them. But the other reason, and as we did touch on yesterday, is that the Inflation Reduction Act makes this topic much more politically relevant or policy relevant than it had been in the past. Before we were just talking about it. Now we have $19.5 billion that are going to be spent on the basis of what conservation practices mitigate climate change. So that brings this question much more into the fore and relevant, I think, to all of us. So this is just their slide. On the right, I like how this is the NRCS slide that has, dollars going into all these climate smart mitigation activities. It covers the various NRCS programs. The last one, the 300 million for measurement, is certainly intriguing to me. I don't know much about it, but this got me looking at it a little harder. But we know there's a lot of billions of dollars that need to be spent, and this is the key list. They talked about it a little bit yesterday. I wasn't sure, but we didn't actually look at that list. It's the Climate Smart Agriculture and Forestry Mitigation Activities list for NRCS, and that's what they're using for deciding how to spend that $19.5 billion. Just to zoom in a little bit on what is in this list, you see the codes and the conservation practices, and that both for the regular codes and then the conservation stewardship program activities. I didn't count how many there are, but it's maybe 4 pages long, something like that. Alan Gillespie last year, for those of you who are in Maryland, he showed us this and talked about it and asked us to contribute studies on the climate impacts of conservation drainage practices. As far as I know, nobody did, because we kept sort of checking in, and because these are really hard questions. In the meantime, and I didn't include this on the slide, but the industry, I can say the Conservation Drainage Network Growth and Adoption Committee, led by Tom Christianson, has really been pushing this. What research can we contribute to this important question? So first of all, I need to mention what the conservation drainage practices are of interest. And so to define this, okay, so what we have is a little diagram that shows free drainage, and many of the speakers will come back to this. So what we're doing is adding practices on to a standard free drainage field. So like bioreactors, we have how it changes these various movements, the various pathways. So we've got the nitrous oxide and the methane from the field itself, and then the nitrate and the dissolved nitrous oxide, and then indirect emissions. So I'm just introducing this here, but many of the speakers will come back to it as we talk about saturated buffers, constructed wetlands, right? We add the wetland in there, and drainage water recycling. I wanted to say this to make it very clear that this is not a presentation on the climate impacts of installing drainage. There's been great work on that. Mike Castellano has talked at a couple meetings, and he has shown potential benefits of drainage itself, especially if people reduce nitrogen applications as they can do with nitrogen. But I wanted to make it clear that's a separate question. Here we are adding the conservation drainage practices to an already drained field. Okay, so again, because of all this question, I agreed to convene a panel of experts to discuss. So at that point, I started looking around who in the country, well, in Canada, has published on this topic. And these 10 people all got together on a Zoom call maybe a couple months ago. Most of them are familiar to people here. They've been to a meeting and spoken at it, except the last two. And I wanted to mention that Eric Davidson, who's really one of the country's leading nitrous oxide experts, and his student Jake Hagedorn had put in an experimental site in Maryland on drainage water management or controlled drainage. So I was very surprised that we were there. Tim Rosen said he does know about it, but they have one of the only published studies on the greenhouse gas impacts of drainage water management. So we all got together, and then one of the outcomes we decided, well, maybe we know enough to write an extension publication. We probably don't know enough to write a peer-reviewed publication, but maybe. So we're thinking about that. So these four signed up to write an extension publication. We've been working on it ever since. Stephen Hall has taken the lead, and I thought we might have a draft here, but it's close to having a draft. And then three of us, so Chandra Madramuthu was not able to be here today, but Lauren Breza from Oregon is also here, as she had contributed a presentation on this topic. So really happy about that. Okay, so now, as I say, I'm going to introduce this panel. Okay, so Stephen Hall, this is his first conservation drainage meeting, and I'm really glad we made this contact. He was suggested to me as someone who knows about drainage because he was at Iowa State as an associate professor for many years, and he is a true greenhouse gas expert among us and does this on agricultural fields. So he's now at the University of Wisconsin, and I was delighted to see he's also an extension specialist. So it's great. We're really glad he's here. Laura Christianson is well known to most people who have been around here, especially she was an associate professor at the University of Illinois for many years. And I think I can safely say she is recognized as the expert on bioreactors, right? That's safe. But she's mostly presented on the water quality impacts and all kinds of other questions. But just very recently, if you may not have seen it yet, her new paper on the greenhouse gas impacts of bioreactors. And she's opened my eyes, and I think to many, of the importance of looking at the dissolved gases, and she'll talk to us about that. Dr. Lauren Breza is a soil scientist at USDA-ARS currently, and so we're really glad that she brings experience from the Pacific Northwest and expertise in soil organic carbon. And Tyler Groh, I've known for many years as he did his PhD and postdoc on saturated buffers with Dr. Dan Janes. And I can remember well all the posters he and Dan Janes' other postdoc gave. And so I was thinking at the poster session last night how great it is that we knew him as a student, as a postdoc, and now as an assistant professor. So maybe that's something the students can look forward to, coming back as an assistant professor and speaking. So before I turn it over to Stephen Hall, our first speaker, I do want to just talk about our sort of complicated agenda, but carefully crafted agenda, and I hope that this will work. Unlike Eileen's and my presentation yesterday, this one has been planned for weeks. So our part one is we're going to talk, these are sort of the background concepts. So Stephen is going to talk about the emissions and how we assess them. Lauren will talk about drainage and soil carbon. And then Laura will talk about the dissolved gases and indirect emissions. These are complicated concepts, but I think they're all great speakers, and I think we'll understand something at the end. And then we will have time for questions and answers just on those presentations, and it gives you a break. I will mention here sure that there are handouts on your table, and please take one. There's a stack, and everybody should have their own handout, if people can make sure that everybody takes one, because we would like you to be writing down questions or thoughts or insights as we go along. Okay, and then you have a couple times to ask them. So that's the background. Then we'll move to part 2, which is the individual practices. And of course, that's what people really want to know, but you have to understand the background. So Laura will talk on bioreactors, of course. Tyler will talk on saturated buffers and constructed wetlands. I will talk on controlled drainage or drainage water management, the practice with 2 names. And Stephen will talk about drainage water recycling. And then we'll have another chance for questions. So we want to make sure that we're interacting throughout this and that people's questions are answered. And then an interesting part, and we'll see how it goes, is we're going to have discussion at the end. So around 10. And then, so as you've been writing your questions and thoughts all along, we have note takers or discussion leaders at every table, I hope. We'll see if we do, otherwise you can manage yourself. And so we'll have some time to talk at your tables, because one of the goals of the Conservation Drainage Network is, of course, networking as well as just listening. So we thought that would be a good chance to for you to talk each other and for us all to get better feedback on what still is unknown and what next steps we see going forward. Okay, I want to stop. Does anybody have any questions? But hopefully you don't have questions at that point. Okay, so without further ado, I'm going to turn it over to Stephen Hall to kick us off.
Well, thanks, Jane. It's such a pleasure to be here and participate in this conference. So I'm going to try to provide some background in terms of what we need to know to actually assess climate mitigation potential of different conservation drainage practices. And I want to start off by saying much of our policy emphasis has been focused on soil carbon, right? We have carbon markets, we have carbon programs, et cetera. But I think we need to be really careful, right? We need to consider 3 key metrics that determine the climate impact of a given practice. So beyond carbon, we have to think about nitrous oxide and methane emissions. And this is especially important in the context of conservation drainage because we're altering moisture, right, somewhere, whether that's within the field or at the edge of the field. And both the emissions of nitrous oxide and methane are microbial processes that are highly sensitive to moisture. And we'll get into the details in a second. Nitrous oxide in particular is oftentimes emitted through what we call incomplete denitrification, and that turns out to be very sensitive to moisture. Methane tends to be emitted when soils are wet for sustained periods of time. Okay, so why don't I include CO2 in the list above, right? We all know that CO2 is the dominant driver of human-caused climate change. Well, CO2 is a little bit tricky in ecosystems, right? It turns out that if we're trying to measure CO2 exchange directly, we run into issues because about half of the CO2 that's emitted from soils is actually produced by living roots. So essentially, they're short-circuiting. They're taking up carbon through photosynthesis. It's coming right back out. So don't worry about the details. The upshot is that we really need to concern ourselves with the amount of organic carbon in soil and how that's changing over time. So, we know carbon cycling is complicated, but at the end of the day, the amount of organic carbon in our soils is really just the balance between carbon input from plant growth, right? We grow plants, those residues decay. Some of them are lost back to the atmosphere as CO2, but some of it remains, right? And it turns out, we know a number of different practices can affect our carbon stocks, but this is challenging to measure. You all are probably aware how heterogeneous soils actually are. And so it turns out to be a complicated endeavor to actually detect changes in carbon over time. Typically, we need really well-designed experiments, we need intensive sampling, and we need to wait a long period of time after practices are changed. So in addition to carbon, right, we have to consider these other 2 greenhouse gases, nitrous oxide and methane. So as I mentioned, N2O is an especially potent greenhouse gas that's mostly produced by microbes, predominantly through nitrification and denitrification. So from an agronomic perspective, typically we could ignore N2O, right? This is a small fraction of the overall N budget. On the order of 1 to 4% of fertilizer nitrogen inputs are typically lost as N2O, right? Sort of a rounding error, right? Difficult to measure. However, N2O packs a large punch in terms of climate. And we can get at this using a metric called CO2 equivalence. How many folks are familiar with CO2 equivalence? Few? All right, great. Well, I'm just going to briefly review this, right? And so what it is, this is an approach developed by the Intergovernmental Panel on Climate Change, and it gives us an apples-to-apples comparison of the climate impact of different greenhouse gases, where CO2 is our reference. And so what we do is we quantify the mass of N2O or methane that might be emitted by a given cropping system, and we multiply it by what's called a global warming potential value. And these values are determined for a given time scale. And we need to do this because once we emit gases to the atmosphere, they have different lifetimes. It turns out that if we emit nitrous oxide today, we're going to be stuck with it for about 100 years before it's eventually destroyed in the atmosphere. And so that gives you a sense. And so over the time that gas is in the atmosphere, that kilogram of N2O is going to have about 273 times the warming effect as 1 kilogram of CO2. Similarly, over 100 years, 1 kilogram of methane is going to have about 27 times the warming effect of 1 kilogram of CO2. So small amounts of N2O and methane really matter. So throughout this talk, we're gonna call negative CO2e values as a good thing. So this is a situation where either we're storing some carbon in soil organic matter, or we've decreased the emissions of N2O or methane relative to a baseline scenario. Alternatively, right, there's cases where we may actually be increasing emissions of either nitrous oxide or methane, and so these are going to be positive values of CO2 equivalent. Okay, so inevitably, right, you probably are aware that the two practices that are most commonly discussed from a climate mitigation potential are cover crops and no-till, right? And so we can get some real potential carbon benefits from these practices. And I think this is sort of a baseline that we need to consider when we're thinking about other kinds of practices we might incentivize. So if you look in the literature, on average, we can expect about 800 kilograms of CO2 equivalent per hectare per year if we grow cover crops on temperate croplands. Again, if we look at any given field, there's going to be tremendous variation around this number, and ranging anywhere from 0 to even greater values. Similarly, for decreased tillage, we might expect similar values on the order of 1,000 kilograms CO2 equivalent per hectare per year in sort of soil types that are representative of our tile-drained croplands. Okay, so that's sort of the carbon ledger. These are some fairly optimistic carbon gains that we might expect. On the other side of the ledger, we have to think about sort of business as usual nitrous oxide emissions. And so some work that my lab did and colleagues in Iowa, we've documented N2O emissions equivalent to about 3,000 to 3,700 kilograms CO2 equivalent per hectare per year, year over year from corn and soybean rotations, right? And again, you look at any given year, any given field, the number can be much lower or much higher. But this gives you a sense, right? N2O has tremendous leverage on the climate impact of our cropping systems. And you'll see the same is true when we go to evaluating impacts of conservation drainage practices. In terms of methane, right, our typical cropland soils can be either small sources or small sinks of methane. It turns out there are some microbes that actually like to eat methane, right? And so this is a good thing. If soils are well drained, we might get a little bit of net methane consumption. Under other circumstances, we might have a relatively small methane source, but you can see that these CO2 equivalent values are quite low compared to these other values above. Okay. However, right, we know there are cases where methane can come up. So one, sort of example that may be salient to many of you are, poorly drained depressions like we get in much of our region. When those are flooded for periods of days to weeks, we can get large methane emissions. So certain parts of the landscape can be relevant methane sources. And especially some practices like, water quality wetlands or bioreactors can potentially produce methane as well. Okay, so what about the climate impacts of our current systems, right? You all are probably aware that when we take forests and prairies and we till them and we drain them to convert them to cropland, right, we've lost a tremendous amount of soil organic carbon across our region. But I think a really important point to consider is that the impacts of differences in our contemporary drainage systems are much less clear. Simply because we have some croplands with higher or lower drainage intensity doesn't necessarily correspond to differences in carbon. And Lauren is going to dive into this in greater detail. I couldn't resist showing one slide from one of our studies in north central Iowa, where we looked at a number of different fields with differences in drainage intensity. And what was really striking is we see no differences in organic carbon, despite really substantial differences in hydrology, simply due to the intensity of tile drainage of those systems. Okay, how about nitrous oxide? So unfortunately, nitrous oxide is complicated. And one of the challenges is that the emissions of N2O are oftentimes nonlinear when we think about variability in soil moisture. And so these are some results from incubations we did in my lab with sort of two typical soil series of north central Iowa. And what you notice for both of these is that the N2O tends to peak when soils are wet, but not totally saturated, right? And so this makes it a challenge. All else equal, we expect as soils are sort of wetting up, drying down, wetting up, or staying wet sort of beyond field capacity, we're likely to maximize emissions of N2O. However, right, this is also complicated because the response is not necessarily the same in every soil, and it's not necessarily the same with depth. So if we're bringing up the water table from below, we may see a very different moisture response function, which my colleagues will touch on in just a second. And finally, I thought I would share some results from a literature synthesis that my grad student and I published a couple of years ago. And so we looked at all of the N2O emission data we could find across sort of the Corn Belt systems, and we found that actually the best predictor of cumulative N2O emissions was the drainage class of the soil series where that experiment was conducted, right? And so you see both for corn and soybeans, when we compare well-drained soils with increasingly poorly drained soils, on average, we increase the annual nitrous oxide emissions. And I want to reiterate, this is an observational study, right? This is simply drainage class. We're not accounting for how intensively tiled each one of these studies actually was, but I think it's really indicative of the role of moisture and covarying properties like particle size and organic matter content, and et cetera. It sort of gives us a place to consider how alteration in drainage intensity may alter nitrous oxide emissions. So we're going to come back to this later in the talk. So I've probably talked for too long already, so I'm going to hand this off to Lauren, but please come and find me or send me an email.
Thank you. All right, let's get situated here. All right. Hi, it's great to be here. My name is Lauren Breza. I am a soil scientist with the USDA in Corvallis, Oregon, and I'm going to continue this conversation about drainage impacts on soil processes. Specifically, I'm going to talk a little bit more about soil carbon, not so much about emissions, but how to keep the carbon in the soil. And I hope to offer a slightly different perspective than what y'all might be familiar with. I live in the Willamette Valley. It's not the Midwest. We have a very different climate and cropping system. And so I hope that you can get a slightly different perspective on things from the Willamette Valley. Okay, so show of hands, who's familiar with the Willamette Valley? Okay, a few people, great. So those of you who are not, it is an agricultural hotspot, and our main commodity that we grow there is grass seed. So if you go to your big box store and pick up a bag of turf seed, 99% chance that all that seed was grown in the Willamette Valley. And the grass seed farmers that I work with often, when we have conversations, I hear all these buzzwords about soil health and carbon stocks and carbon markets and carbon credits. And at the end of the day, when we kind of cut down to what they're really trying to ask us about is, okay, well, are my management practices keeping soil carbon in the soil, or are we depleting our soil carbon with our management? And so how do we build soil carbon with agricultural management? I think some folks might be familiar with the four soil health principles, and those generally just touch on having deep rooting crops and cover crops and organic amendments. And the nice thing about the grass seed cropping systems is that we are perennial in nature and just inherently lend ourselves to fulfilling these soil health principles. So we have deep rooting crops, we have cover crops in the non-perennial years. We leave a lot of residues and organic amendments on the fields. We have reduced tillage, diversified rotations, and we even have a little bit of livestock grazing during certain parts of the year. Okay, so that's how we build soil carbon. Well, I think an important question that the farmers ask and that we should understand is how we lose soil carbon from agricultural practices. So how do we lose carbon? We just do everything I just said in reverse, more or less. But it's actually a very multifaceted and complex issue, and these different management practices can interact with one another to create carbon loss. So tillage and high soil disturbance, low cropping diversity, nitrogen asynchrony, so more nitrogen applied than the crops can actually use, biomass removal without replacement, and I think probably one of the important questions, or I guess management practices relevant to this conference, is drainage limitations. So what do I mean by drainage limitations? I mean using drainage systems or lack thereof in a system where it's not entirely the optimal choice. So that is what I mean by drainage limitations. Okay, have some water. All right, so this is a little cartoon, and I'll explain it in more detail in a second, but what I want you all to do now is to kind of think to yourself as I continue with this talk and discuss these different ideas and concepts and hypotheses, how it relates or does not relate to your specific system and drainage strategies that you use. So just think about that as we continue on. If you have ideas, write it down in that paper that you have. So back to my cartoon, this little, I don't know if you can see my cursor. Can you see that? No, not really. That's okay. That's all right. So this little graph in the bottom right-hand corner, this is just the precipitation graph that we have in the Willamette Valley. So we get all of our rain between the months of October and April. It's a bunch of rain. We have very poorly drained soils, and we rely very heavily on tile drainage to mitigate those high water tables. So this cartoon, oh, does that work? Oh yeah, that might be awkward. In these systems, we rely on tile drainage to remove that water from our fields. So this cartoon just references that. So this is my grass seed system, and moving forward, I'm going to talk about drainage in the context of a grass seed system. So we have high water tables in the winter, very, very low water tables in the summer. In the wintertime in a drain field, as you know, that reduces the water table, but in the summertime, those water tables are very similar regardless of drainage systems in place. So the main question that we asked with our research is, what is the fate of soil carbon in response to tile drainage? So there are two opposing hypotheses that myself and the group that I work with came up with, and one of them Stephen kind of touched on briefly. And that first one is, okay, well, lowering that water table with drainage can create aerobic conditions that have optimal moisture for microbial activity. And this microbial activity then increases the mineralization of existing soil carbon. So these microbes are chomping away at all that organic carbon, and they breathe like just like you and I, and release CO2. The other hypothesis is, well, with that lower water table and deeper roots and increased microbial activity, we will get increased turnover of organic matter inputs that can then become stabilized within the soil matrix. Okay, so how do we do this? How do we test these hypotheses? We sampled up and down the Willamette Valley, which is the ancestral land of the Kalapuya, Malala, and other native peoples. And the Willamette Valley has over 300,000 acres of tile drainage. 31% of all this cropland is drained. And then in addition to that, we have heavy clay soils, which happen to also be very high in soil organic carbon that has not been depleted through these agricultural practices. We sampled in two different regions within the valley, the North Valley and the South Valley, both of which were sampled at two different times of year, so the North Valley in the springtime and the South Valley in the August. We sampled from new tile, old tile, and untiled fields. And these different old tile and new tile just refers to 5 years, less than 5 years post-installation, and old tiles greater than 10 years post-installation. We started tiling the valley in the early 1900s. We have some tile drainage systems that are 50 plus years old that are still functioning and still use the clay tiles. Okay, so I'm just going to show you the total carbon stocks from this project. We've measured a bunch of other things. If you're interested, happy to talk about it later. So we have our North Valley and South Valley. Remember, they are sampled at very different times of year. So one has very, very dry soils, one has optimal soils. So on our x-axis, we have our new and old and untiled treatments, and then total carbon stocks on the y-axis. So in the North Valley, we found no statistical differences between these different drainage treatments. You can see that there's a slight trend that the new tile might have slightly lower total carbon stocks, but that is not a significant result. But I do have some ideas about this trend, and I think it's kind of interesting, so if you want to talk about it, again, happy to talk later. The South Valley, again, no differences in the total carbon stocks between these different drainage treatments. You'll notice the bars are a slightly different color. It's just because we sampled these particular soils based on horizon rather than depth, and you can see the little conversion off to the right of that legend. So in summary, just an overarching summary of this work is that we haven't found any differences in the total carbon stocks in our soils between these different tile drainage treatments. Okay, so why is that? We just mentioned these two different hypotheses. Okay, we could get loss through respiration, we could get loss or stabilization through plant and microbial inputs, but obviously something else is happening because we're not seeing any response to drainage, right? One idea is that maybe we have, our soils are saturated with carbon. If you're not familiar with carbon saturation in the soils, it's a topic for another day, but that is one idea. But as we've been thinking about this, we've come up with two additional hypotheses as to what could be going on here. And the first one is that, okay, maybe we're getting simultaneous loss and stabilization of soil carbon, and the magnitude of these processes are fluctuating temporally. So at the end of the day, we get a net zero change of total carbon stocks. And the final hypothesis, and this actually kind of came about through conversations with Stephen and other folks on this panel, is that, okay, well, maybe moisture doesn't protect the mineral-associated organic matter, but it does protect the particulate organic matter. So our soils are mineral soils, and we can actually fractionate them out into different, to determine where the carbon is being stored. And 85 to 90% of the carbon that in our total carbon pool is actually stored in the mineral fraction. So it's in this very protected fraction of the soil. And the remaining 10 to 15% is in this particulate organic matter fraction. So maybe the moisture is can't really get at that larger pool of carbon that we have. All right, so I just talked about tile drainage. I talked about the Pacific Northwest. I want to come back to this thought experiment that I offered a few minutes ago. What does this mean for soil organic carbon in conservation drainage, and how does it relate to your specific system? And so I think to leave you with some more ideas is that if you have a system that has been depleted, the soil organic carbon has been depleted in, I think it's important that you build that soil organic carbon through these different soil health practices that we just talked about, and then protect it with the appropriate conservation drainage methods that you require for that particular soil type and for your particular cropping system. If you have sustained soil organic carbon, so for example, in the Pacific Northwest, in the Willamette Valley, we have sustained carbon, so we have not depleted our carbon stocks. So it's really, really important that we continue to maintain those soil health practices and keep that soil in place, or soil organic carbon in place, and also protect it with the appropriate drainage practice. I will make a note that we actually don't use any conservation drainage in the Pacific Northwest that I'm aware of. So that's also another conversation worth having, but I just want to leave you with those two things. I think I might be out of time. Quick acknowledgements of the people involved with the project and our funding sources, and I will turn it over to Laura.
Thanks, Lauren and Stephen and Jane. The next part is introducing a couple new concepts. Like most of you, many of you, I am used to taking water samples and thinking about water. The thing I hate about gas samples is that when you take a gas sample, it doesn't look like anything. So frustrating. But I am actually going to introduce a concept that links those two sample bottles. The concept that gases dissolve into liquid. And so it's weird, still weird for me to think about that the water cups on your table actually have dissolved gas in that water. But the concept of gases being dissolved in water is not actually that foreign of us, for those of us who are in the Mississippi River Basin, because we frequently talk about the levels of dissolved oxygen in the liquid of the Gulf of Mexico. That's the hypoxic zone, low DO in the Gulf of Mexico. But of course, oxygen is not the only gas that dissolves into liquid. And in fact, nitrous oxide and methane are greenhouse gases, can also dissolve into liquid, and the quantity of any gas that dissolves into solution is just controlled by those 4 environmental factors. So bringing that back to agriculture and tile drainage, when we talk about nitrous oxide and methane, when Stephen and Lauren were talking about these, we're often talking about those gases coming up from the surface. But what we understand much less is how much those gases are dissolved in the tile drainage water itself. So I want to do show an example of some research we did. In Illinois, we monitored nitrate and dissolved nitrous oxide from these 4 treatments at replicated drainage plots there in Illinois. And the treatments themselves don't focus on, but what I want to highlight is that the order of the treatments, what treatment was high and what treatment was low, was the same for nitrate, which is the graph on the left, and the amount of dissolved nitrous oxide that was leaving the plots. A big difference between those 2 graphs, is the units. So there was 1,000 times more nitrate leaving those plots versus dissolved nitrous oxide. That's kilograms versus grams. We also measured the amount of nitrous oxide coming up out of the soil surface from those plots. And the thing here to note is that the treatment order, or the trend between the treatments, was not the same for the graph on the right versus the graph in the middle. But both of those are nitrous oxide. So I guess my point with this little study that we did was that the graph on the left, the nitrate, we've done a good job as scientists over the last 40 or 50 years understanding how much nitrate is in our tile drainage. We're starting to do a better job of understanding the graph on the right, the amount of nitrous oxide coming up out of our soil surface, but we still really don't have a good handle on how much dissolved nitrous oxide is in tile drainage water. It may be small though. Remember we talked about grams versus kilograms. And so if dissolved gases in water, if those losses of dissolved gases in water are small, do they even matter? And that's where the idea of indirect emissions of nitrous oxide downstream come into play. So this is the second concept that I want to introduce. Nitrate that's in our water that goes downstream gets converted into nitrous oxide as that nitrate moves downstream. The nitrate in the water gets denitrified in our streams, and then in our rivers, and then in our estuaries, and that denitrification produces nitrous oxide downstream. So the current state of the science for how we estimate those nitrous oxide emissions is using what's called an emissions factor. And this is just a ratio, the mass of nitrous oxide that is produced from nitrate that goes downstream divided by the mass of nitrate that goes downstream. And this is used by the IPCC. This is coming from them. This emissions factor has 3 components, starting with the first component, which is emissions from nitrous oxide emissions from groundwater. And that's estimated again by the IPCC using a ratio of, for every kilogram of nitrate in groundwater, 0.006 kilograms of nitrous oxide gets produced. Now, if you measure dissolved nitrous oxide in your drainage and nitrate in your drainage, you can estimate, you can come up with a number for that yourself. You don't have to use this estimation from the IPCC. The second part of the emissions factor from the IPCC is nitrous oxide that's produced in rivers. So for every kilogram of nitrate that goes downstream in a river, 0.0026 kilograms of nitrous oxide are produced. And then the third component is nitrous oxide produced in estuaries. And that's the same number as the rivers. When you sum those 3 components, you see that IPCC estimates for every kilogram of nitrate we send downstream, 1.1 kilograms of nitrous oxide will be produced somewhere downstream. So for us, from a conservation drainage perspective, if we were to send 100 kilograms of nitrate downstream, that would result in 1.1 kilograms of nitrous oxide downstream. If we're doing conservation drainage practices and we send only 10 kilograms of nitrate downstream rather than 100, we end up with 0.11 kilograms of nitrous oxide downstream. It's just estimated using a ratio, and that's important for us to understand. That's it.
Okay, well, thank you. What great presentations, all of you. And we've been working on this publication for a long time, so I've learned a lot, but I learned even more today. So thanks, those were just fantastic. So I don't know where you all are at, and if you have any questions at this point, we wanted to pause and see if there are, and I think we've got our microphones the same as yesterday. If anybody has any questions, I realize that some of you may be thinking, okay, yeah, I'm here for the conservation drainage. So we're gonna get to the practices, but as you can see, it's very subtle, and all these factors play into whether there's a net positive or net negative of any of these practices. Okay, no questions? They're all waiting for that. Oh, Charlie, okay, yes, please. See if it's on. There's a switch in the middle, sort of.
Thanks, Keegan. Morning. I don't really understand these processes very well, but I think
It's really hard.
that the N2O emissions are governed to a degree by temperature. And so I'm assuming that when it's colder, I think I know that when it's colder, you get less, in colder, wet soil, you get less emissions than you do in wet, warm soils. And I'm assuming that it also interacts with the carbon, and most of that is in the top region of the soil profile. And so when you start to think about a combination of subsurface drainage systems with drainage water management, and then what happens downstream, it seems like you have your best opportunity to create a really great production system that's environmentally sensitive, is to have a well-drained, intensively drained field to reduce N2O emissions during the growing season, and then drainage water management to hike that water up during the fallow season, and also make more water and nutrients available to the crop, to the root zone during the growing season. And if that's true, then why aren't we including subsurface drainage as a climate smart practice to have us come in and actually replace a lot of these aging and failing and undersized drainage systems in view of the new climate scenario?
So we're actually going to get to this, and you teed me up very nicely for my subsequent
Okay,
spiel.
I'll stop.
Yeah.
I get wound up. I'll just stop and let you
So I can't resist talking a little bit about the temperature sensitivity. So one of the very interesting observations that's been made over the past decade is that about a third of the N2O emissions from our region actually happen during freeze-thaw cycles, essentially in February, March, maybe April in some areas. When soils are thawing and freezing again, we get bursts of N2O. And this is a really interesting aspect that the scientific community doesn't totally understand, but it's sort of, yes, generally we have greater N2O when the soil is warmer, but then we have this other mode of production that happens during freeze-thaw. And that's sort of another can of worms because we don't really know yet what controls those freeze-thaw emissions. But yeah, we'll get back to that drainage intensity question later on.
Yes, and I'll just mention from the policy or point of view, that would be up to NRCS if they could possibly include it. So we can't speak on that. And again, it wasn't our mission, but Stephen did propose this practice. I only said drainage water recycling, but he actually proposed another one's called enhanced drainage with drainage water recycling. So that's on there as a separate practice. But wow, great question, Charlie. Anyone else? Hard to top that question. Okay.
I'm Ehsan Ghane with Michigan State University. For Lauren, question. I may have missed it. How you had the graph with the tile. How
Yes.
many years was old there?
So the old tile systems were basically 10 years post-installation and older. Most of those systems, I mean, we had a couple that were 15, 20 years old, but actually most of them were around 40 years old.
So I remember, I think just want to mention a point that I think I captured from your presentation. For my soil physics course at Ohio State, Ratan Law, one of the key things that he would teach me in that class was that, yeah, drainage in the black swamp, when we put those in, carbon went out, because it burns out and it goes away. So that's something is kind of, it's concrete in my mind. And I think the point is, one thing that you mentioned in your talk is that it's the reference. If you compare to the Black Swamp, where they had a lot greater concentration of SOC, so organic carbon, then the change because of the reference being so high. So in your case, you showed that some of the, it could stabilize, and some of the reasons that it could actually stay at that level. It's very interesting, though.
Yes,
I think that's all organic carbon.
Yes, I agree. Yep, so the Willamette Valley, we used to have wetlands, and we have no way of knowing what the original SOC content was. So I imagine that there probably was a decent flush of carbon lost when they initially started tile draining in the early 1900s, but we have no way of really knowing what that reference carbon was. I think we could probably reference some of the intact wetlands, and it'd be interesting to actually compare the emissions coming off of our ag fields versus our wetlands, and seeing if our reference carbon is much different in the ag fields versus the wetlands. Yes, interesting question. Thank you.
Yes, great questions. Okay, thank you. And if you have others, of course, you have a sheet to write them down on, so do be thinking about that. Okay, so we're now going to go on to the section that maybe you're all waiting for, is practice by practice, what do we know? And we're starting with bioreactors.
Okay, I'll talk about bioreactors, of course, and a study that Miss Ann Brunton did for her master's in the University of Illinois. She measured both surface emissions and dissolved nitrous oxide and methane at 5 bioreactors in Illinois. The 5 bioreactors were a range of ages, a range of drainage treatment areas. They were a range of designs. 3 of the 5 were designed by the NRCS according to the Conservation Practice Standard. 3 of the 5 had soil covers. And so our point with this study was to measure greenhouse gases at a range of bioreactors under a range of conditions to really get a bead on what is the greenhouse gas impact of bioreactors operating in the real world. Most of the greenhouse gas produced at bioreactors is in the dissolved form. So you can see why I wanted to introduce the topic of dissolved gases. This is important because 5 or 10 years ago, when we were measuring greenhouse gas emissions at bioreactors, we were only measuring what was coming up out of the surface. So most of the story here is in the dissolved form, what's actually happening with the greenhouse gases as that water moves through the bioreactor. Bioreactors work via denitrification, and nitrous oxide is a natural step or a natural byproduct of the denitrification process. So it is unreasonable to expect bioreactors to not produce nitrous oxide. It is reasonable to expect bioreactors to produce some nitrous oxide. But when you look at the amount of nitrate removed, and note those units are grams of nitrate per cubic meter of bioreactor per day, and when you look at the amount of nitrous oxide produced, note those units are milligrams of nitrous oxide produced per cubic meter per day. And when you look at those numbers in relation to each other, you see that on average across all 5 of our bioreactors, less than 1% of the nitrate removed was emitted as nitrous oxide. 0.5% of the nitrate being cleaned from the water left the bioreactor as nitrous oxide. That's efficient. That's efficient denitrification. So when you scale up to the watershed scale, this efficient denitrification on site is really meaningful. We talked about emissions factors, which is just a ratio. And so when you look at the bottom scenario with no bioreactor present, you send more nitrate downstream when you don't have a bioreactor there. And using that emissions factor, which the IPCC uses, you end up with more nitrous oxide downstream when you don't have a bioreactor there, because the emissions downstream are just based on the ratio of how much nitrate you sent downstream. Of course, you also have to consider those increased on-site emissions, but even when you sum those 2 numbers in the top and those 2 numbers in the bottom, bioreactors have the potential to provide a nitrous oxide benefit when you assess emissions at the watershed scale, even when you consider on-site emissions. Bioreactors can also consume greenhouse gases from the environment. Now, we were not the first to show this. Others have shown this, and so I thought that we would detect consumption of nitrous oxide or methane from the environment in maybe 1 or 2 sample events out of our 48 samples. This consumption of greenhouse gases from the environment happened a lot more than I thought. And that was really surprising and kind of encouraging, something to investigate further in the future. The main design finding that we were able to make was that bioreactors with a soil cover reduce surface emissions of methane. And this is really cool. That's the bottom left graph. If you have a soil cover on top, you'll have less methane coming out of the top of the bioreactor. Previous research has found a soil cover benefit for nitrous oxide emissions. That's the bottom right graph. We didn't detect that benefit, but other research has, and so taken all together, there does appear to be a benefit of using a soil cover for emissions of greenhouse gases coming up out of the surface of the bioreactor. Next is?
Yeah, okay. I was just going to say again, because I notice many people don't actually have the handout. So I just want to put it in your head. There's a stack on every table of handouts, and everybody should have one. So be sure that you are writing notes, because we'd love to get these back and get your thoughts too. So please do that. Thanks. And now we have saturated buffers.
Hello, everyone. My name is Tyler. I'm going to be talking about saturated buffers first, and then we're going to talk about wetlands. All right, so saturated buffers, greenhouse gas emissions, what we know and essentially what we don't. So saturated buffers being a new practice, there's not much with, sort of water quality is more and more every year, right? But we're getting there, even less so with greenhouse gas emissions. But my talks for both buffers and wetlands would go over kind of a case study that I wasn't directly involved with, what was good about them, what could have been added, and then sort of summary as far as what we're seeing from other literature synthesized for that extension publication that Jane mentioned before. And so this work came from a study published in 2018 from Davis et al. Many of you guys know Morgan Davis, my counterpart as a student at these meetings. So this study looked at 2 buffers, BC 1 on the left here, and then Iowa 1 on the right. Some of you are familiar with those sites. Sampling that was done included both surface emissions, as you see those chamber tops that we're used to seeing by now on these pictures, collecting surface emissions, and then the wells were used to collect dissolved nitrous oxide as well. Dissolved was taken from the drainage box, so the water coming into the buffer, as well as that last well before the stream. So similar to how we collect nitrate in, nitrate out, this is dissolved nitrous in, dissolved nitrous out. In addition, the field was also monitored, as well as the traditional buffer portion. So with BC1 on the top there, you can see that there's another tile coming in that separated the hydrology of the traditional buffer and the saturated buffer. And so greenhouse gas emissions were collected there. And then at Iowa, site 1, that green portion was far enough out that it wasn't affected by the hydrology of the buffer. So the greenhouse gas emissions were measured in that green portion. Some results on the surface. It's clear to see that the field emissions, especially for 2016, were much greater than the buffer emissions. This has a lot to do with fertilization and the timing of fertilization, and that we get a lot of nitrous oxide emissions the few days/weeks after fertilization. So no surprise there, and that was a corn year in 2016 and soybean year in 2017. That's why there's less nitrous emissions that year. The only year that there was a difference between saturated buffer and traditional buffer was 2015 for BC1. So I do want to point that out. And you can kind of see the range of fluxes, as well as they're usually under 40 grams of nitrogen per hectare per day. And once again, a very efficient, not as efficient as bioreactors, but very efficient nitrous oxide production of only between 2.5% to 7% of the total N removed in that system. So we put this all together, including our dissolved and indirect emissions. You can see here that we add up surface, dissolved, and indirect. There's no difference between saturated and traditional buffers. So putting in a saturated buffer is not going to increase how much nitrous oxide we're emitting from these systems. It's only a difference of 0.01 kilograms of nitrogen per year. However, if we do look at if this was a crop field, it would have been significantly higher, right? We'd go up to 14 kilograms compared to almost 4. So definitely a conversion to a buffer would drop those nitrous oxide emissions down pretty far. I also want to thank Stephen Hall for providing this unpublished data, but soon to be published data. Oh yeah. This is soil organic carbon stocks in buffers over, I guess this is with different buffers of different ages, right, of different years after establishment. And we can see here that the years after establishment definitely does impact the buffer carbon stocks in terms of milligrams of carbon per hectare. And whether it's a restored buffer, so it was once cropland, now it's restored to buffer, or a remnant that was never cropped, but it's a buffer, they do follow a nice positive trend line. So look forward to seeing that publication in the future. All right, and so tying this all together, what does this all mean? The green here represents a sort of climate smart benefit of saturated buffers, and the red is a sort of a negative, right? So soil organic carbon accrual, we can expect around 800 kilograms of CO2 equivalents. Nitrous oxide emission has a range between 48 and 250 kilograms of CO2 equivalents. And then indirect, we do save on indirect, right, because we're removing nitrogen, as Laura was talking about. And so we save on that between 18.5 and 246. And then one big question is methane emissions. It has never been studied on saturated buffers, so we don't really know that impact quite yet. All right, now to move on to wetlands. First, I want to talk about there's two different kinds of wetlands in the drainage field as far as the world of Tyler Groh is concerned. We have the watershed scale wetlands, which I view as the Iowa CREP wetlands, right? They drain hundreds to thousands of they're receiving drainage from hundreds of thousands of acres, right? But then you also have those ones that are draining a field, so tens of drained acres. So you see the two examples here. One from Iowa, the one on the left is the CREP wetland in Iowa, and then the one on the right is the wetland I studied in Illinois with Mark David. Some of you may know Mark. He lives on in the meetings with this presentation. So I do want to mention, though, that the sort of caveat here in how to assess greenhouse gas emissions from these systems is that these riparian wetlands, the ones that drain tens of acres, can have some seepage, as you can see on that photo. There's water that's saturating the buffer portion of the stream. So but I would argue that this is the first saturated buffer ever installed. And we did monitor greenhouse gas emissions as well as nitrate removal in that buffer portion. But it's good to know not only tile coming in, but also groundwater leaving the wetlands or groundwater coming from other sources, right? So it's a big complex system. And so this first example here, before we get into some synthesis results, does come from my master's work at Illinois, and I looked at two wetlands in central Illinois, wetland A and wetland B in the Embra River watershed. We did collect greenhouse gas emissions by using floating chambers and static terrestrial chambers because these wetlands received tile drainage water were not inundated all year round. So you had to do both methods and estimate how much area is inundated versus dry, right? And that was, we did nitrous and methane on a GC and then used the LICOR for CO2 emissions. The results are here. There's a lot to take in. We don't have time to go over everything, but I do have some major points to cover here. The majority of greenhouse gas emissions did come from the terrestrial portions of the wetland, not the inundated portions. The largest terrestrial nitrous and methane emissions happened during the last dry down. So that's bracketed by the orange lines there in both years that was studied. The first orange line is when the outlet stopped flowing, and that last orange line was when the wetlands were officially dry. So you can see there's that peak of methane and nitrous from potentially warming soils, as we were talking about before, right? That's what really spurs on those greenhouse gas emissions. Methane inundated emissions were inversely related to nitrate concentrations in the water, and nitrous emissions were positively related to wetland nitrate concentrations, which is not surprising. And once again, we have efficient denitrification, and only between 1% and essentially 3% of the nitrogen removed was removed with that nitrous oxide emissions, right? So a very efficient process still, but not as good as bioreactors. And so if we put this all together, what does this all mean, right? Wetlands, this thing from multiple sources. You can see in the bottom the sources that were used for this conglomerate. So organic carbon, we can expect an accrual rate in wetlands of 890 kilograms of CO2 equivalents per hectare per year. We do have some nitrous oxide emissions, again, we have that range. We do decrease our indirect nitrous because we remove nitrate again, about 25 kilograms of CO2 equivalents per hectare year. And then we do have quite a bit of methane emissions possible for wetlands. So thank you all, and we'll move on to, yes.
Okay, I hadn't realized that Tyler didn't actually do the subtraction and addition for us, but I think you add the greens and subtract the reds, and then you could figure out the overall benefits of each of these, right?
Best case and worst case scenarios.
Yeah, okay, there's that. Okay. So I'm going to talk about drainage water management in NRCS terms or controlled drainage in many other people's terms. This is Conservation Practice 554. And I thought I'd start out with a transforming drainage graphic just so that you see those graphics and know what I'm talking about. But as I said, for this, we did these new really sketches. And as soon as we figure out what needs to be included and how we're going to show increases in a, decreases in a thing that goes up, we're going to send it to an illustrator, and then we'll have more illustrations to share with everybody in your extension work or whatever. But this is what we've got. So the top one is the free drainage that I showed you. That's just a tile line going out, and then you see the indirect. And then the bottom one is how I've sketched drainage water management. We put the control structure in, and that results in a field being wetter, at least for parts of the year. So that's the difference that it makes. There isn't something in between. But what we know for sure is that it reduces nitrate losses in water. Here, I guess I didn't put that on there, but having just done a big literature review, the average is about 50% or more than 10 kilograms per hectare reduction of the nitrate. So that we'll come back to that. As I already admitted, I've never done these studies, so what I'm reporting on is what other people have done, and we found that there have been 3, and then one additional one that never got published, so it's not going to get published at this point, it's clear. So each of these started out, their introduction says, we are measuring these to see if there might be a pollutant trade-off, like if you're reducing nitrate, which we know controlled drainage does, but in the process, increasing nitrous oxide. That's what everyone assumed, and as Stephen pointed out, it's for good reason, because these soils are wetter, there is a real danger. There's a real possibility of that happening. The first one was in Ontario, and this, some of you may remember Mark Sunohara, who used to come to these meetings from Ontario. They were doing a lot of studies, and he usually reported on the water quality, but they also did this greenhouse gas study, and they found no significant effect on nitrous oxide or methane. Now notice they weren't measuring the dissolved. These are emissions from the field. The second one was in Missouri, and guess who's the author? Kelly Nelson there as third author, who continues to amaze me about the many, many things he studies. So I haven't discussed this with Kelly, but there it is. They also found no significant effect on nitrous oxide. And the third one is this most recent one that I told you about, where a leading nitrous oxide researcher said, boy, we better look at drainage water management. I thought that was very interesting. They also found no significant effect on nitrous oxide or methane. I'm putting one sentence here of explanation because I thought that might give us a clue for future research, those of us who do this. They noticed that the practice raised the water table at the drainage ditch edge, but there was no statistically significant effect on water-filled pore space, or the saturation level at the field soil surface. So we've been thinking a little bit that maybe drainage water management certainly makes it wetter, raises the water table, but maybe it doesn't get wet all the way to the surface. And Stephen tells me that it's really just that surface that makes a difference, right? It doesn't matter lower down. Oh, maybe. All right, we're still thinking about that one. Anyway, for those of us who have collected soil moisture along the way, like with the corn cap, there may be soil moisture data that we could do something with to relate that. But all 3 of them found no significant effect on these greenhouse gas emissions. And this is good news. That's what we, good news for the possibility of drainage water management being a mitigation practice. So it would be very risky to say that we think that it does reduce overall emissions because of the lack of any measurements of dissolved. That's what we haven't done until, Laura is making her case to show that that's a really important thing to measure, and no one has. But basically, if we look at these different pathways, the direct nitrous oxide emissions, if that's no change or non-significant increase, because in fact they did find an increase, but it wasn't statistically significant. The methane, people who measured it found no change. So those are basically at zero. Then there is that reduction in nitrate loss, very consistent. We know that drainage water management reduces the loss in water of nitrate. And then, as I pointed out, this is the big problem. Nobody has evaluated the dissolved nitrate. I don't know that there's any reason for drainage water management or controlled drainage to change the dissolved N2O, but we can't say that, so nobody has measured it. But if that's the case, if no change, then there would be a reduction in indirect nitrous oxide, zero for everything else, and there would be a net reduction in greenhouse gases. So at this point, I think we're able to say that it's likely, although not conclusive. Steve is always cautioning us how much we can say, but this is where we'll continue to think about it, reviewing and making it clear what we know and don't know.
Thanks, Jane. Okay, back to drainage water recycling, right? Most of you are familiar with this. So it's the idea that we're going to establish a reservoir at the edge of the cropland. We're going to capture a substantial fraction of that tile drainage water in the reservoir and then apply that back to the crops during periods of moisture stress. And I think here is maybe where the nuance of how that water is applied could become very important. So if we're using overhead sprinkler irrigation, well, actually, I'm getting ahead of myself here. So first of all, I can pontificate a little bit because to my knowledge, there haven't been studies that have explicitly addressed this practice. So I'll first talk about carbon, right? We wouldn't necessarily expect drainage water recycling to have an impact on carbon, right? We might get a little more plant biomass production, but we're not sure if that might also be counteracted by changes in decomposition. In terms of the N2O, as I was just mentioning, if we have overhead irrigation, if we're delivering nitrate-rich water to the soil surface where most of the carbon and most of the microbial activity is, we might expect that actually to increase nitrous oxide emissions. Whereas, Jane was mentioning, if we're bringing the water up from below, essentially there's less carbon available to drive denitrification. We know that denitrification still happens, but if we're keeping that water table at a constant depth from below, more of that denitrification might be complete. Again, this needs to be looked at in greater detail. So sub-irrigation might actually be better from a climate perspective if we're able to keep this nitrate-rich water away from the zone where microbes are most likely to turn it into nitrous oxide. Okay, indirect emissions. Sort of by definition, if we're capturing that water and keeping it on site and returning it back, by definition, we've decreased the indirect N2O emissions. However, we need to think about this reservoir, and this is where these nuances of how much of the denitrification is emitted as N2O becomes really important. So we need more work there. We can also potentially get methane emissions from these reservoirs. So again, another nuance, but it really depends, as Tyler was mentioning, how long these reservoirs are flooded, et cetera. So some nuance there. So finally, I'm going to get back to the question that I was teed up for earlier, and I think there's some intriguing possibilities for thinking about modernization of our drainage systems and combining this with drainage water recycling. So in places where producers are putting in pattern tile to replace sort of legacy random tile systems, this might have some interesting impacts. So here, let's see, again, impacts on organic carbon, not necessarily well understood, right? There's some reason to think that we're not going to get a huge change in organic carbon, even if we modernize the tile drainage in some of these systems. As Lauren was mentioning, we might have a loss of carbon, but it might also be counteracted by increased plant growth. N2O emissions, as I was mentioning earlier, right, if we accept the idea that drainage and soil moisture is one of the dominant controls on N2O emissions, if we're reducing the average soil moisture values in the topsoil, this may actually help us decrease nitrous oxide emissions from the soil surface. So again, as we talked about from the previous case, if we're holding water on site and preventing it from going downstream, we're likely to decrease the indirect N2O emissions, but we also have to watch out for what's going on in these reservoirs. We might be producing some methane. So again, no one's actually studied this. We'd love to do this study. So hopefully it'll happen over the next few years. So again, sort of to wrap up, so coming back to this synthesis that I showed earlier, right? We know that drainage characteristics of soils have something to do with nitrous oxide emissions, but the details really matter, right? At what depth are we altering moisture? How does that co-vary with other soil properties? And I think this is one of the areas where we really have to do some more work. But to sort of sketch out the importance, right? We talked about sort of the business as usual case for carbon sequestration by cover crops and no-till. To put this in the perspective of CO2 equivalents, if we're able to knock off N2O emissions just by 2 kilograms of N per hectare, this is a similar climate benefit as long-term adoption of cover crops and no-till. And the key point with those practices, the benefits likely saturate over time, right? There's a finite amount of carbon that our soils can store, yet we're going to continue producing N2O year over year in our cropping system. So I think this N2O mitigation really deserves more study. So just to highlight a few research gaps, and again, this is sort of contentious even among our teams, so we were sort of revising these up until the very end, and so, like to reemphasize that relatively few studies have combined rigorous measurements of emissions from the soil surface with rigorous measurements of dissolved gases. So I think this is really important to sort of close the loop on these practices. Also, relatively few studies have thrown carbon into the mix, right? We need to look at all of these together. And I think collectively we've examined some of these practices in specific geographies where we work, but relatively few studies have examined these practices across multiple sites. So if you look at the no-till and cover crop literature, you see tremendous variability, right? We're only able to use these average values because we have dozens of studies, and we're not quite there yet. I think it's a little bit tentative to make recommendations based on one study at a single site or even a couple of nearby sites. But I'd like to highlight that we're really poised to tackle these questions. Got a great team of folks working on these questions. We just need funding. So happy to kick it back to the Jane.
Thank you, Stephen. And I just want to add that these specific research gaps are not contentious, but I was afraid that people would leave and say, oh yeah, it's a bunch of researchers, and the conclusion is more research is needed. So can we do anything more useful than just saying more research is needed? I hope that it's been very clear what we do know and where more is needed before NRCS can make those very important decisions about, the place of these practices in the $19.5 billion. We're really trying to keep that decision in mind as we look at this. So you've seen a lot, and we're way ahead of schedule. Somehow we were all so clear and brief and efficient. So we do hope that you have some questions. And this could include people online. Alan, if you're there today, it was so great to hear you yesterday. Alan Gillespie from NRCS. We would love to have questions from folks that are not here as well. So we'll take some time, see how this goes. You can ask about any of those specific practices, and then we'll move to the discussion at your tables after we've addressed questions.
All right. Burning question. This one's for Tyler. How horrible would it be to collect floating chamber measurements from snowmelt water?
Snowmelt, where in the landscape?
Like anywhere that, because we have a lot of flooded water, but usually the water is, freezing cold. So, that's more what I'm thinking, like in terms of, you folks in the back, the ARS techs in the back will appreciate this question.
So I would say not hard. The one thing is, and I don't know if you saw the picture of me sampling, I was standing on cinder blocks. I had a cinder block bridge going out.
Okay,
because you can't muck up the water. If you muck up the sediment, it's just going to bubble up more and more, gas emissions, right? And so you're getting an incorrect
So you're not actually
flux.
standing in the water.
No.
that's
So,
good.
so if you can make some sort of bridge and like dissipate your weight, yeah, it can happen anywhere as long as the lip of the floating chamber isn't touching the sediment too.
Right. Okay.
Don't disturb the sediment, you can do it. So
Cool. All right. Do it without
And then get
harming
dissolved,
any
too.
students or technicians.
yeah.
No, that's the first. That was my first question. Thanks, Tyler.
Yep.
And then I had another comment. I actually have an alternative hypothesis for the drainage water management, the controlled drainage question. I mean, I agree that I think that you're not affecting at the soil surface. The other thing, that we started to see, we didn't see a lot of changes in microbial response, Haney tests, those sorts of things on Minnesota's coolest drainage plots. That's a study that's published in Ag and Environmental Letters, Sherbein et al. Sorry, quick pitch, pre-tenure faculty. But, one of the things that we hypothesized in our lab group is that there is a lag time in microbial response between when you implement these changes and when you start to see, changes in the emissions in the microbial communities in that capacity. So that would then say if you're changing controlled drainage, that's on a short enough time scale that you wouldn't necessarily be, changing the microbial response, changing the communities as well. I don't know, curious your thoughts.
Yeah, no, that's really interesting. I think that's sort of at the, what the kinds of things we need to address. I will just say the capacity to produce N2O is like widespread across microbial taxa. So there are bugs there that will do it. And essentially we know those details may matter, but I think it's, there's sort of a camp that says, well, really, if you know the physical environment, you can predict what the microbes can do. And we know that works a lot of the time. And again, my microbiologist colleagues get mad at me for saying that, but it's sometimes you can ignore the bugs. Yeah.
Good morning. Again, Mark Wilson, Land Stewards. Question for those of us who do a lot of soil sampling and grab sampling, water samples, can you recommend protocols for testing for these dissolved nitrous oxide methane, and how common is it? I'm not aware of some of the soil labs that I work with that they test for these things. How common is that?
I'm happy to. Yeah, so essentially we can't do it on soil samples that we collect from the field, essentially, which is really a shame. For the dissolved gases, to my knowledge, it's not commonly available at analytical labs. You have to be very careful with how you're collecting the water samples. It's sort of a different methodology than our traditional water quality sampling, and that's really a shame. I think there's the capacity to build infrastructure to make these measurements more routine, but it's not really there yet, unfortunately.
Remember the $300 million. I don't know if anyone here knows where that's going, the measurement and monitoring, $300 million, but that strikes me as maybe building up the capacity for doing the measurements that really need to be made is something that they would be interested in. Did somebody else, do you want to say anything? Okay.
I snuck by both of them. Oh, we had some online questions. So Mark Dietrich asked, is it a zero-sum game for greenhouse gas with both large and small wetlands, or is there a difference there? And then I also had a question for Laura after that one.
Okay.
What happens if we start putting bioreactors on sites that aren't nitrate limited? I know we've kind of had the conversation, like our current standards, like kind of pushing bioreactors towards smaller sites, but if we shift that to bigger sites, what happens?
So the wetlands, so there's not enough data to tease out, large versus small wetlands at this point. But what I will say is that of the studies that did look at like nitrous, this is just for nitrous, right? Not methane, because that's its own beast. But the nitrous oxide emissions can be similar to a crop field. So if you're, converting crop field to wetlands, right, you kind of get that. It's kind of a zero sum for that part, but then you're also removing nitrate, which is the indirect, right? And that gets at the whole where it could be climate smart, because you're removing nitrate here, so it doesn't have to be removed in the stream, estuary, or ocean eventually. So And it's an efficient process within the that system. What we should mention is that denitrification in the stream, right? We don't know the percent of end removal to nitrous oxide emissions, right? We do know for bioreactors it's very efficient, and for saturated buffers and wetlands it's also very efficient. So yeah, it's still good for wetlands. I think as the story in general, especially when you add in soil organic carbon, but we can't really tease out the specifics between the watershed scale wetlands versus edge of field wetlands at this point.
I forgot the question. Something about scale. Larger scale versus nitrate limited. Okay, so we worked really hard to use our 5 sites to try to improve design guidance for bioreactors to minimize greenhouse gas emissions from bioreactors. Unfortunately, all 5 of the sites operated so independently and so uniquely, it was difficult to come up with real design guidance because they each almost had like a different signature in terms of nitrate, nitrous oxide, methane. One of the important things to minimize nitrous oxide, you want good nitrate removal. When we had the highest nitrate removal rates, we had the best nitrous oxide removal also. So keeping the primary purpose of bioreactors in mind of wanting good nitrate removal, that can help minimize nitrous oxide. We did have one bioreactor, one of the 5, that was consistently nitrate limited. It consistently removed nitrous oxide in almost every sample event. It provided a nitrous oxide benefit. It should have produced a lot of methane and didn't. You see what I'm saying? Like each of the 5 sites were very unique. So I know I'm concerned about nitrate limitation, and that's a good reason to scale bioreactor technology. And so that's something I think we need to think about. But our bioreactor that was nitrate limited consistently removed nitrous oxide too, which again was a surprising finding. I don't know if that was your question, but that's a lot of info.
Okay, I'm going to add one thing. Asan, go ahead. I was thinking there wasn't another question. Go ahead. Yeah.
Question for Tyler. But first, this has been a really fascinating panel. Thank you so much. I've learned so much. So Tyler, where did you measure those greenhouse gas emissions on the saturated buffer?
Yeah, so the greenhouse gas emissions, are you talking about the surface ones or the dissolved?
The surface.
The surface, yeah. So they were done in transects as well, right? So I think there was 3 or 4 transects of multiple rings in each transect, right? Similar to like what we do with wells, right? We get nitrate removal. We also wanted to do, so if we assume the highest nitrate is near the distribution line, right? And we're removing as we get closer to the stream, we wanted to get that transect as well. So it's a transect study.
So the reason I asked that question is that I learned from Steve and Lauren is that soil moisture, relationship with soil moisture and wetness of the soil is key with nitrous oxide. So the research for saturated buffers shows that when we monitor the water table in the buffer, we see that the water table is closest to the surface right above the distribution pipe. Yeah. So distribution pipe, water comes out and then it rapidly drops and it goes down. So you would be looking at 5-6 feet. So based on what I learned, I'm not surprised. So it shouldn't be any problem. It's really deep. Water table drops and it goes to the ditch. So probably maybe the place that could be more focused, future research is above the distribution pipe. It's closest, maybe more applications. So did you look at the relationship between the water table and the gas?
Yeah, a little bit. We didn't find very much because the water table where we can monitor those wells, right, we're so much lower because, like you said, the water only stays at that foot below the surface, right by the distribution line. We had a chamber there, like closer to it, right, not right above it, but closer to it. But as far as monitoring groundwater levels, it was within those 3 wells in the transect, right, and that was, a third of the distance each way, right? So we didn't have one right there by our first chamber top.
Yeah, so maybe we need to move one right above it. Yes, yeah. Thank you so much.
Thank you.
Trying to make this quick. Thanks very much on behalf of Tom Christianson with the chair of the Growth and Adoption Committee requested that we touch base on this. Thanks, Jane, for organizing the panel. What a great discussion. I really found it interesting. Quick question. If we added subsurface drainage to this list and considered that to be a conservation practice included in the conservation drainage group of practices, and we had to think about these practices and the effects of them as they relate to the Inflation Reduction Act and climate smart agriculture for mitigation and in greenhouse gas emissions reductions, to get them on that list so they could be applicable for that funding. And also looked at them from a best bang for the buck in terms of N2O or greenhouse gas or carbon equivalent reductions per practice. Where would you put your money?
Stephen, do you want to say anything about that? I'll just say, I think, and maybe someone from NRCS wants to speak up. I just know that since 1985, NRCS has not funded field drainage, right? It's just as a supporting practice for a certain thing. So I don't know if that's a possibility, and that's why we didn't include it in this list, because we didn't think, we did the ones that were the standard. And I'll just say, what I was going to mention, because we had time, was that Chandra Madhur has done extensive work with controlled drainage and sub-irrigation. And so in the extension publication, we will include that, but because it's not an NRCS standard, I didn't actually include it in this presentation for that reason. He has found basically the same thing as what I reported from the other studies. He's got many papers on it, but the conclusion overall is there's no significant difference in the nitrous oxide emissions, and of course there is a decrease in nitrate loss. So just like controlled drainage without sub-irrigation or drainage water management, you would have that. So I wanted to mention that if anyone noticed Chandra Madhur name and contribution. So we were focusing on what we thought NRCS might support, and so it may be more of a policy question than a scientific question, but let me see if anybody wants to answer it from a scientific point of view. Not Laura. Okay.
Yeah. So the question is which of these practices gives us the biggest CO2e? And let me actually raise one other important question I think we have to deal with it. One of the unique aspects of the edge of field practices is we need to consider the area that the practice occupies as well as the field area that it's treating. And so we sort of glossed over this, but we'll get at this in the extension publication. So some of these questions actually depend on that ratio of cropland area to practice area, and the math changes because you can produce methane from a wetland, but if it's treating a very large drainage area and removing a lot of nitrate, it may actually come out ahead, whereas if you have a wetland that's just treating a small field, it may not be such a great climate smart practice. And so this is where the nuance becomes incredibly important. I'm a big fan of the practices that have multiple sort of benefits, so insurance policies, and that's one area where I think the saturated buffers really shine because you're gaining a surprisingly large amount of carbon in the soil. We were, the data that Tyler showed were just sort of hot off the press. This is a master's student that just finished up. So we're accumulating lots of carbon in soil. If you're growing trees, you're potentially accruing carbon in that biomass. You're removing a large amount of nitrate, potentially decreasing indirect emissions. And so you've got multiple sort of insurance policies, especially if you're treating a large drainage area. So I think all of those sort of nuances come into play. And I think these ideas of sort of enhancing drainage but then treating that water or storing it and applying it back through sub-irrigation, I think that's also attractive because we sort of have multiple control points where we might be able to make mitigation opportunities. Anyway.
Okay, thanks for that, and we'll just have to keep working on the drainage, but it's wonderfully described to us. Okay, so now we're right on time for the table discussion, and many of you have been asked to be a discussion leader or note taker. 5 people or so, okay? So if you are willing to share from your group, please come up to the microphone now and be ready for it, okay? You're thinking about it. Okay, John, thank you.
All right, so we had a question about bioreactors. And so with thinking about the full carbon footprint and the CO2 equivalent, if you have a tree, that's a long-term carbon storage. If you put it in a bioreactor, that's going to cycle carbon faster. So what's the life cycle assessment between a tree and a bioreactor? Does that question make sense?
Maybe. We'll have to think about it. So it's just raising questions here. We're not answering questions.
Yeah.
So is there another group that wants to share? And you're going to turn it in, right? Everybody's going to turn in what they had. So some other people, please go to the microphone. We just have like 3 more minutes. So Asan?
So our group, the question came up that how did the wildfires in Canada last summer affect nature and agriculture here? So there was some talks about, because if you notice, there were a lot of aerosols in the atmosphere at those times, June, July. And we were talking about, and then they were saying that they observed higher crop yield, better crop farming in Canada. So we're talking about the possibility that some studies have shown that the Clean Air Act has reduced sulfur in the soil. So some sulfur deposition, potentially those need to be looked at, nitrogen deposition, and also the aerosols in the atmosphere reflect light up. So potentially lower ET, and less drought, possibly. So these are very exciting things to look at.
Okay.
Because we may have another, that may be a routine summer thing, wildfires from Canada. We should get used to that. Yes.
Okay, thank you. Yes?
Hello, Ishkat, landscape architecture grad student. Our group talked about selling practices, specifically stakeholders, farmers, investors, and making sure that they're win-win solutions. And then the second main point that we discussed is how do we contextualize climate smart technologies within climate change trends.
Yeah, thank you. Questions or insights or next steps?
We're a bit of both.
Okay.
Chad Klotzbach, LE&E Services. We, overall, we kind of came up with, how do we quantify the unintended consequences to identify practices in specific locations? So if we have the data, is there a way to, create a specific matrix of, this is here and setting it up that way? The other question that we had is, how do we effectively communicate this data beyond industry, beyond this room of, we all get excited with it, but how do we, here's the paper, are you going to read a 4-page document of the research of it, or can we make it to where other people outside of this room understand it?
Is 4 pages too long?
Well,
okay. I was aiming for 8.
I'll give you an example, right?
But I think it's going to be 12, but I wasn't thinking of 4, but thank you, yeah.
I could have said 40.
Yeah, okay, very good. Thank you, Chad. And I want to call out Katie. I went by there. She's got a full page of notes. I am so looking forward to reading what she has. Thank you, Katie. Yes, Will.
Hi, Will Osterholtz. Our table mostly talked about the difficulties of measuring greenhouse gas emissions and how much uncertainty there is around those numbers, especially given the very episodic nature, sort of the hot moments of emissions. And if there's any possibility of improving those numbers, and then also just if that's a bigger issue with these practices than, generally speaking about greenhouse gas emissions, I don't know. Are these practices, do they lead to more episodic emissions or less episodic emissions?
Yeah, okay.
So that kind of thing.
All right, and you're one who can research it. Thank you. All right, we'll just take maybe one more. And then don't leave without leaving your papers behind if you don't mind. I can share this with the group if you're interested. I'll probably put it on a website where, there's no names or anything, but I don't know if this will form a, research agenda or bring up ideas for next steps, but we're hoping to leave with something, as well as our 4 or 8 or 12 page publication at some point. So, Abby.
Abigail Tomasek, my table was interesting that they were all practitioners, and I thought that their perspective was interesting that in general the message they got was that this is actually good news, right? Like agriculture is not necessarily contributing huge amounts to greenhouse gases. A lot of the messages about agriculture and drainage tend to be painted in a negative light, so maybe some positive news is actually a message that we could spin.
Interesting. I'm not sure it's for agriculture as a whole, but we were trying to spin something positive on conservation drainage. So thank you. And thank you for sharing that. One more. Okay, you're up. And that'll be the last one. And so please leave the paper. So the note takers are taking sort of more generalized notes, but we'd love to see everybody's notes if you're willing to share, unless it's something you really want to take, in which case you could either take a photo of it and take that home, or take a photo and send it to me, I guess, if you want the paper. But let's say leave it on this table up here. Is that okay? Can people work their way up to this empty table up here and leave those for me? Thank you. Last comment.
My name is Babak Dialame from Michigan State University. So we had 2 main questions. how climate change can possibly affect greenhouse gas emissions and the performance of conservation drainage practices. And the second question was, what would be the interactions among different conservation practices and their cumulative effect on greenhouse gas emissions when they are used together?
Okay, thank you. I noticed that nobody suggested next steps, but hopefully you've written some down. We look forward to having the Conservation Drainage Network, and you heard about the committees and all the people who contribute here, or individual researchers or policy people moving this forward. All right, thanks. I'm so glad we were able to do this, and I believe we are on break now until, I don't know, 10:30, 10:40? 10:30, okay. Thank you.